All right, good afternoon, everyone. So today is your quiz day. Make sure you take the time to complete it. You will have until 11:59 tonight to do it. But, obviously don't wait until the last minute. Also, we will be finishing lecture 3 today and also starting on lecture 4 as well. You guys don't have class next Monday because it's National Day for Truth and Reconciliation and the college is closed. So that's why we have to, kind of make up for a little bit of lost time there, because you guys will lose another class for Thanksgiving as well. So yeah, it's going to be a little bit packed uh, with content for the next couple of classes. But as usual, let me know if you have any questions during the class, if I'm going too fast or anything like that. Okay. I have been receiving several emails, people freaking out that they are missing assignments or something like that. Uh, it wasn't just the student at the beginning who asked about it. I got several email saying, "Oh, can I please make up for the lost assignment?" There are no assignments due yet, okay? You will know when things are due if you download the weekly schedule, okay? So I'm just gonna show the class one more time in case you are not sure where to find it. If you go to the course homepage, if you go to content, okay, useful information, weekly schedule. Okay. If you download that, it will tell you exactly when things are due. So you see the Beetroot Lab is not due until October the 2nd, which is like when is that? That would be like next Wednesday. Okay, so we'll have some time. All right. I'll tell you more about it on Wednesday's class. And yeah, you can submit it by then. All right, let's get to it. Last class, we were talking about the endomembrane system, which is a network of membranes that are found inside the cell. It includes things like the nuclear envelope, which extends outward to form the rough and the smooth ER. From the rough ER, you have vesicles that are coming out of it with proteins inside, and then they will move to the Golgi body where it will get sorted, modified, and repackaged, and then it can go to different locations. Right? They could go out of the cell. They might become part of the cell membrane. You might store it in the cell for some time before they're needed, or they might become this other special organelle called the lysosome, okay, which helps break down macromolecules, or sometimes they might release their contents, the enzymes that are inside the lysosome might be released to trigger programmed cell death, or apoptosis as it is called. And so today we will learn about a bunch of other organelles that are found in both animals and sometimes in plant cells as only. All right, starting with this guy called the peroxisome. Peroxisome. Peroxisomes are very similar in terms of appearance with the lysosome. If you look at them under the microscope, this is a lysosome, this is a peroxisome, this is another peroxisome. Uh, I would not be able to tell if they didn't label it on the diagram, right? And I don't expect you to be able to tell either. The point of showing you this picture is to show you that sometimes they look very similar, and yet their function uh, can be a little bit different. Right? So peroxisome, instead of containing enzymes, which is what lysosomes are, lysosomes they have enzymes, right? Contains enzymes which help breaks down various large molecules. The peroxisomes, they don't have enzyme, but instead they have these reactions that are happening in them. They are called oxidation reaction. An oxidation reaction is a reaction where electrons are removed. Okay. And as a byproduct of these oxidation reactions, the the reaction themselves generate these H2O2, which are hydrogen peroxide. Okay, and we don't want hydrogen peroxides to be floating around in the cell. They can cause quite a bit of damage. They could damage DNA, they could damage proteins, and so we just want to contain them within the peroxisome. The function is very similar to the lysosome and to the smooth ER actually. They help break down fatty acids. Sometimes they help break down amino acids. So it's this kind of like recycling depot, and it also helps detoxify poisons. Right? So there's some overlapping in terms of functionalities, but it's like how they do it. That's what distinguish one versus the other. In lysosome, it uses enzymes, and it's part of the endomembrane system. But for peroxisomes, on the other hand, they use these oxidation reactions, okay? And it's not part of the endomembrane system. If you track the movement of a protein produced in the rough ER, it can end up in all the following locations except, so I kind of recap for you at the beginning, right? It can be part of the cell membrane, it can become the lysosome, and it could stay within the cytoplasm as well, right? Or it can be released out of the cell. But it will not be inside the peroxisome because the peroxisome is not even part of the endomembrane system. Okay? So the things that you produce from the endomembrane systems will not end up in the peroxisome. It's kind of like its own thing that is separate from everything else. All right, next we're going to talk about the mitochondria. And this is actually plural, and the singular version is mitochondrion. Right? This is singular. I mean, personally, I don't really care which term you use. I'm just showing you because in case you read about it and then you see like a different spelling and you're wondering why, that's why, okay? Sometimes it's plural when you talk about more than one. If you doing singular, then it's ION. This is the site of energy production. Okay? So you might remember from the second lecture that we produce energy, our cells produce energy through the process of cellular respiration. Okay? So cellular respiration goes something like this. It's time to start memorizing this formula because we will be talking about it extensively in the coming weeks. And it basically takes glucose reacting with the oxygen that we breathe, and you need 6 of them. And you produce carbon dioxide, which we breathe out. You will also produce water in the process. And more importantly, it will produce a ton of energy for you. Okay. And part of this reaction that creates energy happens in the mitochondria. Some cells will have only a few mitochondria, others will have more than 1,000 of them, depending on the energy requirement of the cell. Can you think of an example of a cell that is that's in the body that requires a lot of energy?
Muscle.
Yeah, exactly, right? Muscle. You move your arms and you move your body a lot. So it's not surprising that muscle cells will have a lot of mitochondria. Okay, brain cells as well. Things like fat cells, their function is primarily to store fat. They don't need a lot of energy to create movement, for example. Okay. Now, this is what the mitochondria looks like under the microscope, a real picture. Of course, this is two-dimensional, but the actual thing is 3D. There are a few important structures I want to illustrate for you. Knowing the structure of the mitochondria will be important for lecture 5. Okay? In lecture 5, we will be talking about how cellular respiration occurs and in which part of the mitochondria does it take place. So for now, let's look at the structure. structure of a mitochondrion. So we're gonna draw an oval shape structure like that because that's what they look like. And this here is the outer membrane. Okay, it's a short note, don't worry. Okay, we'll be done in like a few minutes, so you don't have to plan a lot of space for it. So that's the outer membrane. And I mean, if there is an outer membrane, you probably guess that there is also an inner membrane. But the inner membrane is a little bit different from the outer membrane, specifically, like it's not smooth like this, okay? The inner membrane is actually gonna fold up like that. It's gonna fold up, okay, like an accordion. It's gonna fold up multiple times, and it formed these um, finger-like projections. That's how usually textbooks describe it. They're like little fingers like that, and then you can go all the way around and draw the inner membrane. Okay, so these foldings, they are called cristae. Now, let me write it down for you. This is the inner membrane. Inner membrane folds up into finger-like. That means they look like finger projections. Okay, not my words. That's just how they are usually described in textbooks. Okay, finger-like projections called cristae. Which increases, which increase surface area for reaction. Okay. Generally speaking, when you carry out a reaction, such as cellular respiration, reacting with the glucose and the oxygen and so on and so forth, generally speaking, the more surface area you have available, the more efficient you will be at performing the reactions. Okay. You want to be able to create energy quickly and in an efficient manner. That is like no backlog, right? You're going to be able to use up the fuel quickly. And that's why the inner membrane folds up into these cristae. Between the outer membrane and the inner membrane, there is a gap, and this gap is filled with fluid. We call that the intermembrane space. And that space, as you will see in lecture 5, it's very important, uh, as is everything else actually, to the mitochondria. And finally, last thing we're going to add to the diagram here is in the inside of the mitochondria, the deepest part is called the matrix. And there's a lot of things going on inside the matrix, and we'll learn all about it in lecture 5. Okay, so that's it. I just want us to draw together a simple diagram of what the mitochondria looks like, and we'll come back to it in future lessons. Okay, next we're going to talk about the chloroplast. Chloroplasts are found in plant cells and not animal cells. Okay. Um, Remember we talked about there are animal cells, plant cells, and then there are these things called protists, right? Example of protists, we only learned two of them, are amoeba and paramecium. It's in the earlier slides. Okay, so those don't have chloroplasts, but some protists have chloroplasts. Okay, kelp is an example of protists. They have chloroplasts. I found in plant cells and some protists. And not everything is just animal or plants, right? There are protists. There are fungus, right? Different kingdoms. The chloroplast is where photosynthesis takes place. Okay. And photosynthesis is the reaction that allows the plant to make their own food, okay, by using the sun's energy. So I can write the equation for you. Okay. It is they're gonna take carbon dioxide from the atmosphere. They're gonna take water from the roots, absorbing through the soil, and then with light energy, they will convert it into oxygen, which gets released into the air, and glucose, which they store as starch. So this process, if you pay attention, is actually the reverse of cellular respiration. So in cellular respiration, we take the glucose and we react with the oxygen, but in photosynthesis, it produces oxygen and the glucose. In order to capture sunlight, the chloroplast contains this thing called chlorophyll. Okay. Basically, the green parts of a plant, they contain the chlorophyll. And that captures the sunlight and then allow the reaction to happen. Similar to the mitochondria, the chloroplast is double membrane. It has an outer membrane and an inner membrane. You do not have to memorize these names. Okay, that's all I want you to know for the chloroplast. Okay? So plants, they have chloroplast as well as mitochondria. We do not have chloroplast. Now, this is an important diagram. It's a nice diagram as well. That summarizes the relationship between cellular respiration, which is this process right here, taking the glucose and the oxygen, producing carbon dioxide and water. That's cellular respiration. The products of cellular respiration can be used by the chloroplasts to do photosynthesis, right? And then the products of photosynthesis become the reactants for cellular respiration. So these two processes complement each other, right? The ingredient of one is the product of the other one. And it allows like a cycle to keep on going and going and going. Okay. So we breathe out the carbon dioxide. The plant will use the carbon dioxide to make glucose. The glucose is stored as starch. When we eat the starch from like potato, for example, our body burns them and convert them back into carbon dioxide. And then round and round it goes. Okay. So please take a closer look at this diagram when you're studying. It's important. Okay, next we have this thing called the central vacuole. The central vacuole is found in plant cells, and they take up more than half of the cell volume. This whole thing is a cell, is a plant cell, and you can see the central vacuole takes up quite a bit of space. What you will find in the central vacuole would be mostly water and other nutrients. By storing water in the central vacuole, it creates pressure that would push against the cell wall. And that will allow the plant to stand up and open up its leaves. Right? If you forget to water your plants for a couple of days, right, the leaves start to get all wrinkly, right? Like, or if you don't have plants, if you think about like buying, I don't know, celery sticks or something, and then you leave it in the fridge for too long, then they start to wither up, right? That's basically the central vacuole losing water. Okay? Without the water in it, there's no pressure pushing out. Everything just kind of shrivel up. Okay, kind of like a balloon. If there's no air in there to create the pressure, it wrinkles up, right? Sometimes the central vacuole can store other things like pigment. Pigments are basically colors for the flower petals, or in some cases, they might even contain poisons that helps protect the plant. Okay, I mean they don't all have poisons, but you know, in a plant that does make poison, like poison ivy, for example, then you would expect to find some poison in the central vacuole. Anybody have any questions before we move on? Excellent.
But excuse me, do you
Sure.
have do you have vacuum in animal plant, animal cell?
We do not have central vacuum in animal cells. Yeah. All right. Central vacuole is not the only kind of vacuole. Okay, there are other vacuoles that exist. It's called contractile vacuoles. These are found in some protists, okay, single cell organism, e.g., the paramecium. You don't have to memorize the name of these things, okay? But I bring this up every now and then because it has a lot of features that we want to talk about. So this paramecium it's a protist. Okay? A lot of protists, they live in fresh water, like a pond, okay, a river stream, something like that. And one of the challenges in living in these freshwater environment is that water will constantly be coming into the cell. And if the cell does not have a way to remove these water, it will swell up and burst and die. Right? And so the contractile vacuole basically are pumps. Right? It fills up with water and then it gives it a good squeeze, and then all the excess water that have came in will be expelled. Okay? So it has to keep on doing this. Otherwise, it is going to die. Okay. So we don't have contractile vacuoles. We, as in like animals, and plants did not have them either. Okay. It's like a special feature for some protists.
I have a question.
Sure.
Can we say that this central vacuum is part of the homeostasis of parameci
It's
or
it's part of the come again.
homeostasis?
Oh, homeostasis. Yes, exactly right. It's a way to maintain internal balance. Otherwise, it's gonna burst and die. Very good.
Uh, is a physiologic or behavioral?
That's a good question. Not everything can be defined as physiological or behavioral, right? Because this is a very simple organisms, like they don't really think, right? So it's hard to define in this case. Good question though. All right, next thing we have is a plant cell wall. A cell wall is made up of cellulose, something that you should know for today's quiz. And unlike the cell membrane, the cell wall is not selectively permeable. It lets everything through. Okay. The main function of the cell wall is to protect the plant from bursting. Okay. As the water is coming in to fill up the central vacuole, it's like a balloon getting bigger and bigger and bigger. Okay. It likes it that way, though, the plants, right? I told you it helps it stand up against gravity. But having a wall there around the cell really gives it some protection from all these high pressure. Okay. So we don't have cell wall. Uh, Plant cells have cell wall. Other organisms like fungus, mushrooms, right? They actually have cell. Mushrooms are not plants, by the way. Okay, mushrooms are fungus, and they also have a cell wall, although it's not made up of cellulose. It's actually made out of chitin, right? But yeah, it's for protection of the cell. All right. Select all the false statement below. All right, let's do a poll and see how it goes. Okay, it was that's almost a minute. But I only have 7 people answering so far. 8 now. Okay, so I'm going to end the poll now, and we will take it up. Okay, so first one, smooth endoplasmic reticulum is part of the endomembrane system. That is a correct statement. We're looking for the false one. Okay, again, endomembrane system includes nuclear envelope, rough and smooth ER, Golgi body, and lysosome, right? So if you can remember them, make sure you make a list for yourself. There is an abundance of mitochondria in muscle cell. That is true. Abundance means a lot of them, okay? Muscle cells needs a lot of energy to produce movement. So not surprising they have a lot of mitochondria. All cells have cell wall to protect itself from the environment. No, that is not true. We animal cells we don't have cell walls. Hydrogen peroxides are found in lysosome. That is also incorrect. It is found in the peroxisome. Finally, chlorophylls are found in chloroplast. That is true. Okay, chlorophyll is what makes the plant green, and it absorbs sunlight. Okay, if you think of vegetables that are darker green color, they have more chlorophylls, like kale, spinach. Well, these things are very rich in chlorophylls. Nutritionally speaking, they don't do anything for us though. Okay, like there's not much nutritional value to chlorophyll. Okay? But they are there in the dark green vegetables. Okay, we are moving on to the last part of the lecture, which is cytoskeleton. Okay, so there are three kinds of cytoskeleton I want you to know. It could be a little bit confusing, overwhelming, but just try to understand the overall theme here, and maybe it's gonna make it a little bit easier. So skeleton, if you imagine, like think about the word skeleton, they are like our bones, right? Like in the body, the skeleton provides a framework. It supports our body, right? It protects things, right? So these are cytoskeleton, which is kind of like skeletons, but inside the cell. Okay, so each cell has a network of these cytoskeleton that overall will help provide support, just like the real skeleton, and in some cases allow movement as well, also like the actual skeleton. So there are three types of fibers here that makes up the cytoskeleton. I mean, there are more, but these are the three kinds that I want you to know. The first one is called microfilaments, which look like these things. There are microtubules and intermediate filaments. I know the names themselves are already confusing. So, I encourage you to make a table when you're studying, put them nicely in your help sheet and with the functions beside it, and that should help you remember it a little bit better. So let's start with the microfilament. Okay, the microfilament, uh, it's on the next slide, but I just want to point to this picture here. Okay, if you focus on the microfilament here, it's actually made up of two strands. Okay, two strands that are like twisted together, braid together, intertwined together, and each strand is made up of these little balls. Yeah, each ball is called an actin protein. So you take a bunch of actin proteins and you link them together, you get this long strand. And if you take two of these strands and you twist it together, then you get yourself the microfilament. Right? A filament is basically like a long rod of some sort, okay? Long fiber of some sort. So if we staying a cell, okay, like there are different techniques that you can do to a sample of cells. And one thing you can do to them is you can highlight specific proteins that you're interested in, okay? You can actually highlight specific regions of the cell that you're interested in looking at. And then you can make them fluoresce, okay? Make them glow and give these nice green color, red color, and whatnot. So in this particular picture, you can see this is a cell, okay? And this technique that they are using to highlight the cell, they are highlighting the nucleus, so you can see the nucleus as a reference to where things are. And all these green things that forms like a web that goes throughout the cytoplasm, these are the microfilaments. Okay? Like they are not actually green. People get confused all the time. Like the cell is so thin, like if you look at it under the microscope without any treatment, everything is like see-through. Okay? But these are special techniques that we can use to highlight different parts, different features of the cell. So if you highlight the microfilament, you can see that it's like all over the cytoplasm. And they actually provide attachment sites for various organelles. Right? So it turns out, the mitochondria, it's not just like floating around randomly. Right? Think of this as like a Velcro board, and you can stick things onto it, right? Okay, so put the mitochondria here, right? Okay, put the rough ER here, and they are kind of anchored in place by this web. Okay, or like think of it as like a spider web. Things get stuck to it. They don't randomly move anymore. Okay, provide attachment sites for organelles. The second function is to enable cell movement. Not all cells in the body will move, right? But many of them do. Take white blood cell, for example. They can move around in the body and find things to eat, right? Bacteria, they will eat them up. Damaged cells, they would eat them up. So in order for the white cells to move around in the body, it needs to use these microfilaments. Okay? Now this is really hard for me to show, you know, through the computer screen, but try to visualize this with me. Okay? Let's say this is a white cell. It's not, but let's just say it is. Let's say it wants to move that way. Okay? So what it needs to do is it will take all these actin, all these microfilament, which are made of those red little proteins, right? Red circle that I showed you, right? So if the cell wants to move to the right, it will start breaking down the little red circles here. It will break down all the microfilaments on the left side. So now we have a bunch of free red circles, a bunch of free actin proteins. These actin proteins will all move to the other side of the cell, and then they will start building more of it. Okay, they will add it onto the right side. So over time, you are shifting the microfilament from one side of the cell all the way to the other, and this kind of shifting motion causes the cell to move forward. Okay, so it's all about relocating the microfilament and creating motion. I hope that makes sense to you. All right. There are three kinds of cytoskeleton. Microfilament is what we just talked about. There's another one called intermediate filament. Okay. Micro is like small, right? Okay, small filament, and now this is like medium-sized filament, intermediate filament. You can see that they're a little bit thicker, and actually they kind of occupy similar space in the cell. Okay. It's also kind of all over the place in the cytoplasm. But these kind of intermediate filaments, they are particularly important in forming intercellular junctions. What that means is in some parts of the body, if you have cells next to each other, they are gonna put a staple. They're gonna put a staple between the cells. Okay, so things are not gonna leak between them. Right? Think of your intestinal tract. Right? That's made up of cells. You don't want food to be leaking out of it, right? It's going to cause all kind of problems. So to lock the cells in place between each other, to seal the gap between them, you need these intermediate filaments. The intermediate filaments are also important in stabilizing the nuclear envelope. Right? We know the nuclear envelope surrounds the nucleus and it protects it, and we need it to be intact. Otherwise, if the nuclear envelope falls apart, then the DNA, the genetic material inside might get damaged. Right? And that's what happens in this condition called progeria. Okay? And some of you might have seen pictures like these before, where there is a person that looks really old, but at the same time, they kind of look like a young person as well. So this is actually a very young person. Okay, it's like 14 years old. But he has progeria, okay, which is like accelerated aging. Okay, and one form of the disease has to do with a mutation in a type of intermediate filament called lemon A. When this protein is not correctly created because of mutations, the nuclear envelope kind of become unstable. Okay? And as such, the chromatin structure is going to be affected, and the gene expression is going to be different. When I say gene expression, it means like, which part of the DNA do you turn on and which part do you turn off. So all that get misaligned, and the outcome is accelerated aging. Okay? There are currently no treatment for this. Partly it's because it's a very, very rare genetic disease. And if you don't have enough people who have it, then you know there's not a lot of money going into researching for a cure, right? But there are some breakthrough recently with gene therapy. You can look it up if you're interested. It is called progeria. Progeria. All right, the last kind of um, cytoskeleton is what we call a microtubule. Okay, a microtubule. So I want you to go back and think about what we learned last class. Okay, you have this vesicle that's coming out from the rough ER, right? Hold on, just want to show you. We have this vesicle that are like bubbling out from the rough ER over here, and it needs to go to the Golgi body. But how does it know where the Golgi body is, right? Like, how does it know to go that way and not like that way, right? Well, it turns out not like the movement is not random, right? Everything that moves around inside the cell, much of it relies on uh, the guidance of the microtubules. Okay, so you don't have to draw it on your diagram, but there is actually a track here. Okay, this track here is a microtubule, and the vesicle can hop on. Okay, and it will transport it to the vesicle. Sorry, it will transport the vesicle to the Golgi, and then it will hop off. Okay. And there might be another microtubule that will take it like elsewhere. Okay. Like after the vesicle comes off, okay, there could be another microtubule that will allow it to travel all the way to the surface of the cell. Okay. Now this is for real. You can do your own research, okay, and look it up. There's a lot of videos of this online. So there is this protein called kinesin. The name is not important. I'm not going to test you on it. But if you think of like drawing a stick figure, okay, but then you take away the head, okay. So this is basically kinesin, okay. It's like a stick figure without the head, and so it has 2 feet and it has 2 arms, all right, and it will carry the vesicle with its 2 arms, and then the feet will actually walk along the microtubules. Okay, and so it carries the vesicle and then it will walk along the microtubule and take it to where it needs to go. If it needs to go to Golgi, no problem. We'll get on this microtubule track. It will take you to the Golgi. If you need to go to the surface of the cell, then you take another microtubule, right? So nothing is random. That's my point. Okay? And it kind of aligns with the theme that I've been trying to tell you, right? Like one of the characteristics of living things is that they have order and complexity associated with them. Right? And that orderness and complexity exists all the way to the cellular level, even within the cell. Right? Nothing is random. Right? Now, microtubules also form other important structures, and I'm going to show you a few of them. Okay? There are these spaghetti-looking things that are in animal cells, not in plant cells. Okay, They are called centrioles. Centrioles. The centrioles, they produce a special kind of microtubules called the spindle fibers. Spindle fibers. Okay, we'll talk more about these things in lecture 7. But for now, the spindle fibers are important for guiding chromosome movements as the cell divides. Now, you might remember one of the four parts of the cell theory states that a cell will divide to create more cells, right? All cells comes from the division of pre-existing cells. And since the cell has 46 chromosomes, so when it divides, you need to first you need to copy the chromosomes, and then you gotta sort them. You gotta be like, okay, you get 46 and you get 46, right? Which means you gotta move the chromosome around. And to do that, you need the spindle fibers. Okay, these spindle fibers are made up of microtubules. Okay, so microtubules allow guidance of vesicles, but they also help guide the movement of other things like chromosomes. Okay, never mind these like kinetochore stuff like that. Okay, like it's not important at this moment. Okay, these are the chromosomes, and these are the spindle fibers that are trying to pull on them. Okay, finally, the microtubules also made up two other things. They make up the cilia and they make up the flagellum. Okay, flagella is plural, but for humans and for many animals, in fact, the only place you will find flagellum is in the sperm. Okay? So the tail of the sperm, that's called flagellum. Right? And it moves in like a whip-like motion that helps the sperm swim. Okay. Cilia, they usually cover the entire cell. Right? So if you think back the paramecium again, the one with the, the contractile vacuole, the protist that lives in a freshwater, they have a lot of cilia covering the entire body. Okay. In fact, I'll go back to show you. You don't have to go back. You can just watch me go back. Right here. You see these hazy, fuzzy line here around the cell? Those are the cilia. And they beat together to help the cilia, to help the paramecium move around. Okay. So the cilia and the flagellum, they are both made up of microtubules. We actually talked about cilia before. I showed you a similar picture as this last time. It was black and white. This is a better one. This is found in your trachea. Okay, trachea is your airway. Right? I told you the cilia, they're gonna help sweep up the, dust particle that we breathe in, right? And then we are able to get rid of them that way. In the female body, there is an other place where you will have cilia. Okay, so these are cilia. Does anybody know what this picture is? This is only in the female, not males. Which part of the body do you think this is that has cilia in it?
Fallopian tube.
That's right. That's correct. This is the fallopian tube. Fallopian tube, or sometimes called the oviduct. It connects the ovary, which is where the egg comes out, to the uterus. Okay? So when the egg is released from the ovary, the cilia will help guide the egg towards the uterus. Okay, so like if the egg is moving and it bumps to the side, then the sweeping motion of the cilia is gonna tell it to, okay, keep rolling, you're not there yet, right? It's gonna go towards the uterus. Okay. And that is it. Okay, this is a summary that tells you things that are unique for plants and things that are unique for animal cells. Okay. Now, some of the stuff we didn't talk about, you can ignore it if we didn't talk about it. Like, for example, we didn't talk about an amyloplast, just ignore them. Okay. These, whatever crystal, forget them. Okay. Like, I cannot find a picture that's like, only have the same things that we talked about. Yeah, so just ignore any things that we didn't talk about. And this centriosome, it's same as centrio. For us, it's the same. Okay. I mean, they're technically not the same, but who cares? Okay. Just keep it simple. Centrioles. That's it for this lecture. Any question at all? Everybody happy? Okay, silence is happy then. Uh, which of the following is made of two intertwined actin protein? Well, I'm just gonna do this one myself. It is the microfilament. The microfilament. Okay, now we are gonna switch gear and talk about lecture 4. And before we do that, we have to take another quick note. Okay, so please take out a piece of paper and we'll do it. Okay, this one is a little bit takes a little bit longer, uh, but it's an important note. Okay, and my program is crashing a little bit. Let me try to restart it. Alrighty, let's see. So we are going to be talking about the cell membrane quite a bit because lecture 4 is about moving things across the cell membrane. Okay, you need to bring things in, you got to send things out. Everything needs to pass through the cell membrane in order to enter or leave the cell. And so we need to look at the structure of the cell membrane. Okay, and the model that people use to describe cell membrane is called the fluid mosaic model of the cell membrane. Okay, I just want to remind you that another name for cell membrane is plasma membrane. They mean the same thing. Okay. Now, fluid means it is not solid. Okay, we don't want the cell membrane to be like a wall behind me, okay? Because if it's a wall, then it's completely solid. Nothing can pass through. Nutrients can come in, waste product can go out. We do not want a system like that. Okay. We want it to be fluid. Okay. So there is um, some dynamic movement to it. Okay. It's more like a soap bubble, if you would, right? It's not static. Okay? So that's what the fluid part means. Mosaic means like if you think about a mosaic window, it's like a one of those window that you usually find maybe at like a church or like a mosque, where the window is it's made up of like different color glass, right? So like you have some yellow, some blue, and so on and so forth. This is called mosaic. Okay, so what that means is the cell membrane, the composition of the cell membrane is not uniform. Okay, it's not one thing. Instead, it is multiple things that are kind of combined together, and they kind of connect together to form this fabric that is called the cell membrane. So that's what the mosaic model means. All right, let's draw the cell membrane then. Okay, so here's our cell. This is the nucleus. And we are going to take a slice of the cell membrane and we are going to zoom in and take a closer look at its composition. Now, help me out here, guys. Yes, you guys are super quiet recently, but hopefully you can tell me what is the major component of the cell membrane?
Phospholipid.
Phospholipid is correct, okay? I hope the rest of you also had a similar question answer in mind. And so when we draw the cell membrane, we're going to be drawing a bunch of these phospholipids. And remember, the phospholipid has a hydrophilic head, and it also has hydrophobic tails. Okay, now the tails might both be saturated, or sometimes it might be unsaturated. So I want you to draw a bit of both in your picture. Okay, here I'll throw in another one. And it actually is not just a single layer. It is going to be two of these phospholipid layers together. Okay. And for reason I will explain in a bit, when you draw it, I want you to draw, I want you to draw like that. And then I want you to draw another one here. Okay, there's another one. But leave a bit of space here. Okay? Leave a bit of space. Okay? Leave a gap over here. Let me try it one more time. Okay, so leave a bit of a gap, and then I'll do another one here, like that. All right, I'll give you a moment to draw that, and I will tell you more. Okay, do not draw this next part. All right, do not draw this next part. But watch, okay? This is like a cross section of the whole membrane. Okay, you can think of the membrane as like I don't know. There's a different ways you can think about it. You can think of it. Don't draw this. Okay, you can think of it as like a fabric like this. Okay, like a mattress, if you would. And then we're just taking a look at the cross section. You cut it open and you take a look here, right? So that's where the phospholipids are going to be, right? So rather than drawing the whole thing, we're just, taking a look at the 2D view of it. But in fact, you're going to have all these phospholipids like forming the entire sheets, right? Do you understand this? Again, I apologize. It's very hard to convey this in a virtual environment. But if you don't understand, please feel free to ask, because I guarantee you are not the only one if you have a question. Okay, so it's like this whole sheet, okay? But for simplicity, we're just looking at like a 2D view of this. Okay? One way to think about this is you guys know ball pits? I think they're called ball pits. It's like, kids, they sometimes jump into this whole area with balls, right? Those plastic balls. Okay. So think of the balls as the hydrophilic heads. The ball can move past each other easily, right? No problem. Right? And that's exactly what happens with the phospholipids. The phospholipid, like if you think about one individual phospholipid, this guy can go left and right, no problem. Yeah, it can move between each other. So in a 3D sense, if you think about this phospholipid, this guy can like move back and forth, all over the place within the same layer. Right? But it's very rare for the phospholipid to flip to the other layer. Okay, so like if you are on the outer layer, you tend to stay there. Okay? Very rarely it will go to the other side, but generally they stay within their own layer. Okay, so let me write that down for you. Again, you don't need this big diagram. You don't need to draw it, but I want you to be aware of the situation, right? So, phospholipids can move freely within its layer, but rarely will they flip to the other layer. Okay. No flip-flopping. Moving side to side, it's okay. So this whole thing is called a phospholipid bilayer. Phospholipid bilayer because there are two layers. Okay, the inside is made up of the hydrophobic tails, which is thicker than the hydrophilic heads. I told you to think of a peanut butter sandwich, right? The bread or the hydrophilic heads, the peanut butter in the middle, that would be the hydrophobic tails. Every now and then, you will have a protein that's inserted in the cell membrane. Right? So some proteins will go all the way through, like this. Right? Some proteins, they might just be stuck to the inside or stuck to the outside. Okay? So these proteins that goes all the way through, we call them integral. Integral protein. Integral protein spans the entire membrane. Okay? It goes through it. Whereas these that are just stuck on either the inside or outside, we call them peripheral protein. Okay. Outside is also called peripheral. Okay? The difference between the integral and the peripheral is like, is it just on one side or does it go all the way through? Okay. Any questions before this gets more complicated? All right. Now, in addition to the phospholipids and the proteins, you also have these. Oh, by the way, like you might be thinking, like, what are the functions of these membrane proteins, right? We'll talk about it in a second after the break, okay? But remember the pathways of the repackaged proteins, like where do they go, right? One location is to become part of the membrane. And so all these proteins came from the Golgi. Okay? But exactly what do they do? I'll tell you after the break. Okay? Now, you also have these steroids, okay? These cholesterol in the membrane. Okay? So cholesterol, they controls the membrane fluidity. Okay. More cholesterol in the membrane equals to more fluid. Because you're kind of the cholesterols are kind of like getting in the way between the phospholipids. If the phospholipids cannot pack together closely, then it becomes more fluid. The cell membrane becomes more fluid. If you have less cholesterol, then it's more solid. They are able to pack together closely. Not a lot of space between them. They tend to become more solid. Okay. So I'll explain when do you want it to be more solid and when do you want it to be more fluid in a bit. Now, finally, the last thing I want to add to this diagram are these sugar on the proteins. Okay. Some proteins will be modified by the Golgi, where they would add some sugar to them. some glucose to it, perhaps. It's also possible to add the sugar to the lipids. So these are sugar, carbohydrates. So this whole thing that has protein with sugar stuck on them, it has a name. It is called a glycoprotein. And perhaps not surprising, this thing, it's a lipid that has sugar stuck on it. That's called a glyco. Glyco means sugar, okay? Glycolipid. Okay, so glycoprotein are sugary stuff stuck on a protein, and then glycolipids are sugar stuck on a lipid. And these things, the function is part of the function anyways. Okay, they have a lot of functions. I'm trying to just keep it simple for you. And I know what you're thinking. Even though I'm trying to keep it simple, it's already very complicated already, right? So the glycoprotein and the glycolipids, they play a role in what's called cell-cell recognition. Right? How does your body tell which cell belongs to the body and which ones do not, right? If there is a bacteria that invaded your body, your cells have to be able to tell, like that does not belong, right? We got to attack it, right? So these glycolipids and glycoproteins, they form an ID tag, an identification tag on the surface of a cell. And if a cell has something that's different from what you have, then it's considered as foreign, and they will attack it. Okay? No other individuals will have the same ID tag as you, unless you have a twin. Okay, like an identical twin. Okay, uh, otherwise, no one else is gonna have the exact same ID tag. That's why, like, it's hard to find compatible organ donor. Right? Even the best match, it's not gonna be like, perfect match. Right? So you try to match it up as much as you can. All right, time for a break. Let's come back in 5 minutes. We will resume at 1:40.
Professor, before we go to break, can I
Yeah,
ask a question?
sure.
So the assignment is not open yet for me.
Yeah, it's not open yet because I haven't teach you how to do it yet, right? So like
Okay,
I don't
okay.
want people to do the lab before I explain it. So like people don't do it wrong, right? Basically.
Sure, okay.
Okay, yeah.
Sorry.
No problem. Okay, let's continue. Now that was basically the quick overview of some of the things I want you to know about the cell membrane. And now we are going to go back to the lecture here, lecture slides, and see how much we can go through. So lecture 4, membrane transport. Right? We are let's see. So like next, you know what? Actually, we'll talk about that later. Okay, but then the quiz 2 is going to be coming up soon as well, and then there's going to be test 1. Test 1 is going to be lecture 1 to 5. Okay, so we have 3 lectures down, 2 more to go for the test 1 for the first half of the course. All right, so we did the fluid mosaic model. Fantastic. Let's take a look at this question. This is also an important thing to think about. How would the percent of saturated, unsaturated fat, and cholesterol content of a cell membrane found in plants growing in cold environment differ from those growing in hot environment? Okay. To paraphrase, basically, if you look at the cell membrane of something that grows in really, really cold places, what would you expect to see for their saturated, unsaturated fats, and cholesterol content in the cell membrane. Okay. Now, like I said, this is very important. So please pay attention. If you're watching the recording later to hear me say it again, it's gonna be around the 1 hour and 10-minute mark. Okay, so write down your notes. 1 hour 10-minute mark. This is where I will be explaining that. Right? So if it's cold, then things tends to become more solid, right? Things become more solid. Like, I mean, you put water in the freezer, it's gonna turn into solid ice. Okay, so in general, when it's cold, things tend to become more solid, and that includes the cell membrane. Okay, if it's really cold, then the movement of the phospholipids is gonna slow down, and then they tend to come closer and closer together, and then they become more solid. Now, we do not want this, right, for the organism, because if it does become more solid, then the cell is no longer going to be able to exchange things with its environment, right? And so to counteract the cold environment, to prevent it from becoming more solid, you actually want the membrane to be more fluid, more so than, say, in the hot environment. So in order to do that, more fluid means you need to have more cholesterol. Okay? And you need to have more unsaturated fatty acid in those tails. Right? Like more of these bended ones, right? That will help the membrane to be extra fluid. So when it does get cold, it's okay. It's not gonna be solid too quickly. Okay? Are we together? And so I want you to think about what happens when it's hot. And I mean, sorry, obviously here it will be less saturated, right? Less saturated fatty acid. So think about this when you have headspace. I know most of you are thinking about the quiz. I took a look. Only a few of you have did the quiz. So, this might not be the best time to think too much about this, but you definitely should revisit it when you have time. All right, let's talk about the membrane protein function. All right. So I told you there are two kinds of proteins that you can find on the cell membrane. They either insert themselves all the way through, and we call them integral proteins, or they might be stuck on the outside or stuck on the inside. And when they're stuck on the inside, they don't necessarily have to be stuck on the cell membrane directly. They could be like attachment onto the integral protein. Okay? The whole point is, like, does it go through or does it not go through the membrane? Okay? So both kind of proteins will have their different functions, but generally speaking, we can summarize the membrane protein functions into 6 different types of functions. Starting with the first type. They actually provide attachment sites to the cytoskeleton. Right? Let me go back to here to show you. This is our drawing, and you don't have to draw it on yours. I don't want you to ruin your lovely, lovely note. But remember we talked about before the break that we have these actin filament, intermediate filament, and all that stuff. So all those cytoskeleton fibers, they got to anchor to something, right? So when it gets to the edge, when it gets to the cell membrane, they actually attach onto some of these proteins. Okay? They're like little pegs or sticks that anchor, like a camping tent or something like that. Okay, it keeps it in place. All right, so the membrane's not gonna like fly off. Right, so they provide attachment sites for the cytoskeleton, and they also, not only are they useful for the inside of the cell, the membrane protein will also help the cell to attach to the extracellular matrix. Okay, Cytoskeletons are actually not just found inside the cell. There are some similar stuff that we didn't talk about though, but they're found outside of the cell. So this is a cell, this is a cell, this is a cell, and all these fibers that you're observing, that's part of the extracellular matrix. Okay, so if a cell wants to attach to this network, then it needs to do so through the membrane proteins. Okay, so attachments to cytoskeleton. These are red cells, and again, the membrane proteins help it maintain that particular shape by attaching to the cytoskeleton. Okay, number 2 is cell signaling. Cell signaling. Some membrane proteins are receptors. Right? So receptors, think of them as like little doorbells. Right? So special signaling molecules like hormones, they would bind to the receptor, the protein, and then it will relay the signal inside the cell to cause changes inside the cell. And the kind of changes that will happen depends on the kind of signal that is conveyed by the molecule. Right? Some molecule will increase certain types of cellular activity, while others will suppress them. For example, adrenaline. Adrenaline binds to a receptor protein that is located on the cell membrane. So this is adrenaline. Another name for adrenaline is epinephrine. Okay, it doesn't really matter. Like you might have heard of like EpiPen, right? For people who have allergies, that epi is epinephrine or adrenaline, same thing. So they will bind to the receptor, and you think about like a person who's getting ready for like a sprint or something, right? They are gonna need all those extra energy to complete the race. And so the adrenaline in the body is going to bind to the cell receptor, and then it's going to cause a bunch of reactions to happen. Okay, these reactions will ultimately lead to hydrolysis of glycogen. You know glycogen. That's the storage form of energy found in muscle or liver. Right? And so if you're getting ready for a race, then yeah, you want to break down the glycogen, change them back into glucose so you can burn them for energy. Okay? So this is basically a function of the membrane protein. Okay. This always reminds me of a story, and it might help you remember this stuff. Okay. Like some of you know Halloween is coming up, right? Like in a month, a bit more than a month. And when I was growing up, my parents they are anti-Halloween. Okay, something about handing out free candies, they didn't like that concept at all. Okay, so every year when it's Halloween, my parents would be like, you know, we're not gonna open the door, we're gonna turn off all the lights, so we pretend no one's home, right? But you know, kids are gonna ring the doorbell, and every time when someone rings the doorbell, my parents were like, "Okay, go hide in the basement, okay, and don't make a sound." And then we'll all like scurry up in the basement, and then until we hear the people leave, and then we're like, "Okay, we can come back up now." Okay, this is for real. That's what we did. Okay, so this is kind of like that, right? The molecule, the signaling molecules are like the kids ringing the doorbell. Okay? Even though the signaling molecule never physically make it into the cell, even though those kids never physically come into our house, it causes all these changes, all these commotions that are happening inside the house. Okay, it changes the activity in the cell, right? And that's what the receptor protein is. It relates what's going on outside to what's going on inside. Okay. Your neurons actually communicate in a similar fashion, but never mind this. Okay, you'll learn more about it later. It's just an example. All right, so we're talking about different functions of membrane proteins. We already have attachment to cytoskeleton. We have number 2, cell signaling. And number 3, it is enzymatic activity. We already know proteins are sometimes enzymes, and they could help speed up or catalyze chemical reactions. And a lot of times when we have a chemical reaction, it doesn't just convert A into B. and then we're done. It doesn't happen like that, okay? But rather, there are multiple reactions that will happen one after the other. So let's say you have a circle molecule, it will go into the first enzyme, and then it will get converted into this triangle thing. And then this is not the end. We actually need to convert the triangle into something else, into a square, and then convert it into an oval, and finally convert it into this hexagon thing. So you actually need to do 4 separate reactions in order to get to the end product that you need, right? And rather than having the enzymes all over the place in the cell, you put them all right next to each other at the cell membrane. So immediately when you create one product, you could use it for the next reaction, and then you could use it again for the next reaction, and then for the next. It just makes things happen faster. Okay, it's the same reason why, most people would put things that they use together in the same drawer, right? Like I imagine, your kitchen, you would have the knife, you would have the grater, right? You would have the chopping board all within the same area because you're going to be using them for the same task, right? You're not going to keep the chopping board in the bathroom and every time you need it, you're going to go run and get it, right? So for the same reason, okay, if these enzymes are related to the same task, same set of reactions, there's no reason to put them randomly all over the cytoplasm. Let's just all put them all at the cell membrane so you can use them together in quick succession. Okay? Any questions? All right, 3 more to go. Next is the intercellular junction. We talked about this briefly already earlier when we discussed intermediate filaments. There are parts of the body where you have to staple cells together, where they have to hook onto each other so things don't leak between them. Okay? In addition to the intermediate filaments, we also have some membrane proteins that will allow us to form those kind of attachment. Right? So like the intestinal tract, that would be an example of a location where you would need something like this. Blood vessels as well. We don't want the blood to be leaking all over the place. So you're going to staple the cells together. Elvis, do you have a question? I see that you are unmute.
No.
No? Okay.
No. Sorry.
Number 5 is cell-cell recognition. We talked about this briefly already when we wrote a note. Glycoproteins, sugary proteins, and sugary lipids, they collectively form an identification tag. Okay, and the immune system can use these tags to distinguish what belongs to the body and what does not. And here we have a white blood cell. You just have to believe me, it is. And then this is like a cancer cell. Okay. And sometimes the cancer cell might have different ID tags than the ones inside the body. They're very good at hiding it, as you will learn later on, but sometimes they do have different ID molecules, and then your white cells are able to attack them. Okay. When this happens, the cancer cell never becomes like a full tumor. It's when this fails, then the cancer cell starts to divide in an uncontrolled manner, and then the person might get a tumor. So cell-cell recognition. And lastly, membrane proteins are needed for transport to move things across the cell membrane. Right? Some things can move through on their own. Others, they are too big maybe, or they are too hydrophilic. They can't do it on their own, so they need the help of a transport protein. Someone asked the question, how can it fail? How can the white blood cells fail to detect the cancer cells? Well, that's a great question. Short answer to that is, unlike things that invade the body, like bacteria and viruses and fungus and parasites, those things have very different tags on the surface than our own cells. So as soon as they come into the body, your immune system reacts and it gets rid of them. Okay. Cancer, on the other hand, as we will learn later on in the course, they arise from our own cells. Okay. So many times their surface tags are virtually identical to our own cells. So it's very hard for our immune cells to detect them, okay? Sometimes the changes occur earlier on on the cell, so you're able to pick them up earlier. But sometimes by the time it shows some kind of difference on the surface, it's already too late for your white cells to detect it. By that point, it's already kind of out of control. Okay, there's like a point of no return, if you would. I hope that answers your questions. But we are going to learn more about that later on. Okay, back to the transport stuff. I don't have time to go through this with you, but maybe you can write down the answer like E, okay, and then you can try to do it again later. Just write it somewhere secretly, okay, so you don't remember it as E. Can give me a definition of WBC? Yeah, sure. WBC is white blood cell. Okay, white blood cell. I'll just type it out for you. White blood cell. Okay. Red blood cells carry oxygen for you. White blood cells kill bad things in the body, basically. All right, here's another one. Which of the following is not a function for membrane proteins? The answer is D. Okay. Like normally I would do these with you, but the last time only 7 of you answered. So I don't know, maybe you guys are just not in the right mindset for now because of the quiz. So I'll just give you the answer and you can do it later. All right, I am going to wrap this up by talking about one last thing, okay? Which is cellular energy. Cellular energy. Okay, there's another question. I just joined and finish. Yeah, that's okay. Okay, yeah. If you miss, you can just watch the lecture. Yeah, it's okay. No problem. Thank you for letting me know. Where was I? Right, cellular energy. Now, metabolism. Metabolism. This is where metabolism are basically all the reactions that are going on in the body, in the cell. Okay. And at any given time, there are a lot of reactions that are going on. Each reactions are catalyzed by an enzyme, right? That helps lower the energy requirement and allows you to do it at inside the body. Now, we can separate the metabolism into 2 categories. We have catabolism and anabolism. Catabolism, the prefix cata is the same prefix as catastrophe. In a catastrophe, everything breaks apart, right? So that's what catabolism means. Catabolism means you are breaking down molecules into smaller unit. So if you're taking a polysaccharide and you're breaking it down into individual glucose, that would be an example of catabolism. If you're taking a protein and you're breaking it down into amino acid, that will be an example of catabolism. On the other hand, anabolism is building things from smaller molecules. Right? And building things is going to require energy. Okay? Whereas breaking things down, it actually releases energy. Okay? So let's take a look at this diagram. You have it in your study guide. And if you didn't print the study guide, that's fine. You can write down whatever you can and add to your study guide later on. So over here on the left, we have macromolecules. Let me just zoom in here. It's kind of small. Macromolecules from food. Okay, so in preparing for today's quiz, you probably know that we eat things like carbohydrates, proteins, lipids, right? We don't deliberately eat nucleic acid. It's just found in all living things, right? So I'm not going to write that. But these are the macromolecules. They're big molecules. So what do we do? Well, we're going to have to break them down into building blocks. Right? The many building blocks for biosynthesis, for creating other biomolecules. So what kind of building blocks do we have? Well, for carbohydrates, maybe we're going to break it down into glucose. Right? For the proteins, we're going to break it down into amino acids. Okay, maybe glycerol, and like, fatty acids, right? So all these building blocks. So in the process of breaking them down, this is called catabolic reaction. Catabolic reaction. Also known as catabolism. In the PowerPoint slides, it says catabolic reaction releases energy. Releases energy. So we're going to have energy, and the energy is actually going to be temporarily captured by something. We'll talk about that something in a bit. And more importantly, the energy that is captured will be used to take the building blocks and build things that we need in the body. So these are like different from here. These are macromolecules needed in the body. Okay. Maybe we need to build some glycogen to store in the liver, and we're going to use the glucose and we build them. Maybe you need to make some proteins for your muscle. You got to build those. Maybe you need some testosterone, progesterone. You're going to have to build those. Okay, so the building, that's called anabolism or anabolic reaction, and that requires energy. Okay, so basically, what's really important to know here is that the energy released from catabolism drives anabolism. Right? That's why we need to keep eating, to keep on producing energy so that we can use those energy to build things that we need in the body. There are plenty of things we need in the body to build. Right? Tons of proteins. Every time you grow nails and hair, those are new proteins. We don't eat nails and protein. We don't eat nails and hair. The protein that we eat from the meat, for example, they are broken down into amino acids. And from those amino acids, you can make things like nails and hair. It's pretty cool, actually. Now, we are not very efficient in this. Right? If you think about like a light bulb, those really old school light bulb with the tungsten inside, with the filament inside. Okay? Like after you leave it on for some time, you touch it, is hot, right? These are called incandescent light bulb. How efficient do you think those are? Somebody like what percent of energy are actually converted into light? Anybody have a rough idea? Okay, 40%. Anyone else? Just throw me some numbers, guys. We're almost done. 60%, 50%. Yeah, those are great guess. They are only 10% efficient, okay? That's why like if you touch it, it's super hot. Most of the electricity are wasted as heat, okay? So 90% of the energy are lost as heat. And then we have the fluorescent one. The fluorescents are the ones that, that looks like a little twisted thing. Those are about 85% efficient, okay? And then now we have the LEDs. They are about like 90 to 95% efficient, okay? So you touch an LED, it's like barely hot, okay? The body is about 25% efficient, okay? So we do not capture all the energy. In fact, much of the energy are lost as heat. Okay? The body is about 25% efficient, right? Which is fine. I mean, like we are warm-blooded organism. We want to keep the body at 37 degrees. So as long as we eat, we generate heat, we keep ourselves warm. Are we okay? All right. I think I'm just gonna do one more slide and we're done. 2 more slides. Sorry, 3 more slides. Okay, we're almost there. So cellular respiration is a catabolic reaction. We are breaking down glucose. In fact, we're breaking down glucose into carbon dioxide, and part of it is going to happen at the mitochondria. Okay, we're going to learn all about this in lecture 5. It's the most difficult lecture of the first half of the course. Okay, so please make sure you come to class for that. And I haven't really tell you about this yet. I keep on saying energy, right? You're releasing energy, you're producing energy. These energy, you need to capture them, right? Because we don't use all the energy immediately, right? We can capture them, we can store them, and we can use it later. Right? Much like, a battery. A battery is stored energy, right? And you can plug that battery into your electronics and appliances, and you can run it, right? And so in the body, we have a type of battery. Okay? They are called ATP. Right? ATP stands for, it does not tell you what it stands for on the slide. I'll tell you later. I'll write it down for you later. It's adenosine triphosphate. The key thing here is the T, triphosphate. There are 3 phosphate on it, PO4, PO4, PO4. This ATP form, this is the charged version. Charged version, fully charged, 100% charge, like your cell phone, and you can use it. You can use it for different things. Okay? What can you use it for? You can use it to move things, okay? Like move the flagellum. Okay? This is another protist that has 2 flagellum, and you can move it. That's called mechanical work. Right? We can move our muscle. That requires energy. That requires ATP. We can use it to move things across the cell membrane, and we can use it for chemical reaction. Enzyme lowers the chemical reaction, right? Why we talk about that. Lowers the energy required for a chemical reaction to take place, I should say. So instead of using 100 unit of energy to start a reaction, you only need 60 unit of energy, maybe. But what happens to the 40? Nothing is free. Someone has to pay for that 40 units of energy, right? Well, in some cases, it comes from ATP. Okay, those batteries that I'm talking about. Every time you use the ATP, it gets converted into ADP, adenosine diphosphate. Right? You break off a P and it releases tremendous amount of energy. You use it to move things, you use it to transport things, you use it for reactions. And then afterwards, the ADP is like drained battery. Okay? But we can recharge it. Okay? The energy we absorb from the food, the energy that we get when we break down macromolecules from our food, that energy is actually used to recharge the ADP. And once you recharge it, it goes back to the ATP. And then you can use it for other purposes. Right? So the ATP, when you use it, becomes ADP, and then it keeps on charging, using it, much like your phone. You charge it and drains it on a daily basis, right? Except in a cellular level, right? We are charging it not with electricity, we're charging it with chemical energy from our food. Okay. And there's one more question here. I'll just do this with you. Catabolic reactions consume more energy than they produce. That's not true. Catabolic reaction actually release more energy than they consume. That's why we can capture the energy that are released from them, right? They break down polymers into monomers, yes. They produce excess heat to warm up the body. Remember, we are not very efficient, only 25% efficient. They do not just happen in humans. Okay? They happens in other cell type as well. Plant cells do it, protists do it. Okay, so we're not the only one. All right, and that's it. Now, good luck with the quiz today, but Wednesday's class is important. We'll finish lecture 4, and I will also be talking about the lab. Okay, the lab is going to be due next week, so make sure you come to class for that. Right? You want to hear the explanation. No, it's not group, it's individual. Okay, each of you have to submit your own lab, and it's done online. You don't have to go to the school. It's pretty straightforward. Okay, you'll see. You'll see. All right, have a good one, guys. Thank you. Bye bye.
Thank you. Bye.